Hi, my name is Davachi, Assistant Professor of Psychology and Neural Science at New York University, and this is Tambini, a graduate student in our lab and lead author on our recent paper in Neuron. Today we're going to tell you about our recent findings linking resting brain correlations with memory consolidation. It is known that upon encountering, for example, a person holding an object such as this basketball, activation in cortical regions that process those visual stimuli is important for remembering each individual stimulus, but that memory for the co-occurrence of the two is supported by hippocampal processes that serve to rapidly bind together these elements. Now, after this encounter, when attention is no longer focused on the person with the basketball, consolidation theory hypothesizes that the same hippocampal cortical regions that were active replay or reactivate patterns of activity characteristic of the prior experience during offline states to further strengthen or consolidate those memories. Thus, the restructuring of memory is thought to be supported by interactions between the hippocampus and cortex during offline states such as sleep and awake rest.
There is a lot of evidence for hippocampal reactivation during sleep that is related to memory. However, although possible, it is unlikely that sleep is the only time when recent experiences are consolidated in memory. Much less is known about the role of offline awake rest periods for memory consolidation. Recently, reactivation of hippocampal activity has been seen in rodents during periods of awake rest. Furthermore, this reactivation is greater following novel experiences and those preceding reward, both situations in which memory encoding may be enhanced. However, reactivation during awake rest has not been directly related to changes in future memory performance. Furthermore, it is unclear how these findings in rodents will translate to the human. Thus, we wanted to see whether post-experience resting brain correlations in humans are related to later memory performance.
Participants were scanned during encoding of the stimulus pairs and during rest periods interleaved both before and after these encoding sessions. We focused on neocortical regions known to process the individual stimuli we used in our study and the hippocampus, which is known to be important for the initial associative binding. Furthermore, it is interactions between the hippocampus and relevant cortical regions that are hypothesized to underlie memory consolidation. We first examined baseline levels of correlation between the hippocampus and LO, a cortical region known to be involved in object processing during an initial rest scan. Here you can see an example of baseline resting correlations between the hippocampus and LO in a representative subject. After the baseline rest scan, subjects then performed an associative encoding task where they were asked to form a meaningful association between an object-face pair. As expected, during the task, correlations between LO and the hippocampus were high. Critically, we then examined whether hippocampal LO correlations would be enhanced during a rest scan following the task compared to the correlations seen before the task. And we were delighted to see that hippocampal LO correlations were indeed enhanced during the post-task rest period. But are these correlations related to memory? We addressed this in two ways. The first was to look at task differences. Now, we have not yet mentioned that in addition to the object-face encoding task, subjects also encoded scene-face pairs during the scan. When we tested subjects' memory following the scan, we found that associative memory was greater for the object-face pairs compared to the scene-face pairs. Interestingly, as shown on the left, the enhanced level of correlation between the hippocampus and LO during post-encoding rest was only seen following the object-face task, the task that resulted in higher levels of subsequent associative memory. Now, the second way that we addressed the extent to which these correlations are related to memory was to see if the magnitude of resting correlations actually predicted subjects' later associative memory performance. Indeed, as shown in the scatter plot, the subjects who had greater subsequent associative memory were the ones showing the greatest post-task resting correlations. These results provide strong evidence for a major prediction of consolidation theory by demonstrating that hippocampal cortical interactions during offline states are important for strengthening the day's experiences in memory.